First of all, I'd like to sincerely thank Dennis for inviting me to this conference, Annette, the organizing committee, for making this happen, and Sir Anton for allowing me to live in his palace. This is just a great, I still couldn't believe it. It's just an amazing place. I'm an assistant professor at Long Island University in Brooklyn, New York, and my background really is plant evolution. And as I've been telling other people here, I actually feel like an imposter because I don't really have personal experience with psychoactive plants. My love affair with psychoactive plants only began 2 years ago. There was an international graduate student from Saudi Arabia who wanted to study psychoactive plants. She was particularly interested in harmal. And because my background is plant evolution, I told her, okay, so let's look at psychoactive plants at an evolutionary perspective and let's see if we can find any patterns. So this work was published last year, and I'm going to talk about that today. And in addition to that, I will also talk about a very similar study wherein we look at traditional medicinal plants, so not just psychoactive plants, also at an evolutionary perspective. So before I begin the talk, I'd like to also thank my two graduate students who work with me on these two papers that I'll be presenting today, Camila Javier and Nashmaya Al-Rashedi. So without their help, I won't be standing here today. So in 2012, there was this fascinating paper that came out in PNAS, wherein the authors tried to test if traditionally used medicinal plants can actually guide drug discovery efforts using phylogenetic analysis, so evolutionary analysis. And in phylogenetic analysis, so when you reconstruct the phylogeny from sequence data, let's say DNA data, information is assigned to the nodes of the phylogeny instead of one species at a time, which facilitates a study of trait distributions. And so in their study, what they did was reconstruct the molecular phylogeny, so the phylogeny based on DNA sequences of 20,000 species within 3 different regions. So they had Cape of South Africa, Nepal, and New Zealand. And what they found out was that traditionally used medicinal plants were not scattered randomly across each of the phylogenies, but were concentrated in certain nodes. Okay? Certain plant families or certain groups were medicinally important. And they happen to be similar in these 3 phylogenies. So the important medicinal plant family in South Africa was also seen in Nepal, was also seen in New Zealand. And so in their study, they concluded that cross-cultural comparisons at the phylogenetic level can actually guide our drug discovery efforts so that we focus on these traditionally important plant families. And so this study inspired my own studies in herbal medicine. In the United States, herbal medicine is part of complementary alternative medicine, and it has been thriving for the past several years, even decades. So it has gained a resurgence in the United States. It's a multi-billion-dollar industry. You have at least 40% of American adults admitting to using some type of CAM or complementary alternative medicine. If you go to Rite Aid or CVS, like these drugstores in the United States, you will see many herbal supplements. Okay, so or even herbal teas, so people would take them for insomnia or for mild depression, things like that. So it's a booming business in the United States. And in New York City, where I work, there's the herbal medicine industry has been thriving as well. Okay, so this is not surprising because New York City is one of the most diverse regions in the world. You have at least 800 languages being spoken there. More than 30% of the population is an immigrant population. If you go to Lower Manhattan, you see Chinatown. Go to downtown Brooklyn, there's a big Middle Eastern community. You go to I guess uptown Manhattan, like Harlem area, there's an African community. In Queens, you have the Latin community. So it's not surprising that these different immigrant cultures have brought with them their traditional medicine, their herbal medicine into the city. And so for this first study that I'm going to be presenting, we actually focused, and this is in collaboration with my student Camila Javier, so we actually focused on 5 major immigrant groups in New York City. The Hindu or Indian immigrant culture and their Ayurvedic medicine, the Chinese culture and their traditional Chinese medicine, African, Latin, Caribbean, as well as Middle Eastern. So these were the 5 major immigrant groups that we focused on. And our question was, do these immigrant cultural groups in New York City use related plants, evolutionary related plants for similar ailments? Okay? So we tried to answer this question by reconstructing a phylogeny or an evolutionary tree of the different herbal species that are used by these cultural groups. So what we did was we visited different herbal stores in the city, specializing in Chinese medicine, Ayurvedic medicine, Islamic traditional medicine, and so forth. And we compiled a list of about 95 species, okay, from these different herbal stores in New York City. And then we took note of their medicinal applications based on the product label, so if it's used for diarrhea or constipation, respiratory problems, things like that. And from the plant names, we went to GenBank, which is a government database where you can download genetic sequences. So we went to GenBank, we download the sequence for the RBCL gene, which is a photosynthetic gene. So it's universally present in plants, and it's often used in phylogenetic analysis. So for each of the herbal species, we got the RBCL sequence. We used this DNA sequence to reconstruct the molecular phylogeny of these medicinal plant species. And we also indicated on the phylogeny the originating cultures. So which culture is using this particular plant and if it's native to their original region. Okay? And we also indicated the medicinal function. So as I said, if it's for constipation, diarrhea, but we categorize this into these applications. So those that we think fall under gastrointestinal, then we like diarrhea, constipation, it will be under this category. Those that are used to alleviate, say, hypertension, that will be under cardiovascular. Those that are used for, say, decongestion or for respiratory problems, then it will be classified under this category and so forth. So what you see here is a miniature phylogeny from our bigger phylogeny. I'll show the bigger phylogeny later on. And just a quick primer on interpreting the phylogeny. So these were 3 different species that we found in our survey, so when we went to different herbal stores, that are used by different cultures. So the different cultures are indicated by the different colors on the branches. So this is Middle Eastern, red is Middle Eastern, pink is Chinese, green is Ayurvedic, and I'm missing here African, which we indicated with black color, and Latin Caribbean, which we indicated with a blue color. Okay, so for these 3 species, only these 3 cultures are using them. And then on the right side of the phylogeny, we have the medicinal applications. So if we found in the product label and also in the literature, we also looked at the literature, if we found that this particular species is being used for gastrointestinal problems, then we would put a blue box next to it. So that's true for all of these 3 species. So they were being used for gastrointestinal problems by these 3 different cultures. Okay? We didn't find any traditional uses for them for cardiovascular, so this goes in sequence. So the first column is for gastrointestinal applications. The second column should be cardiovascular, but we did not find any traditional use for cardiovascular problems. We did not find any traditional use for respiratory problems and so forth. But then for antiparasitic or antibiotic applications, we did find information. So these 3 cultures were also using it for antibiotic uses. So this group right here, so this belongs to the family Lauraceae. So this is the node that I'm talking about. This is the common ancestor of Lauraceae in this group or clade. And then you see another node right here. So these 2 Cinnamomum species are united together by this node because they are more genetically similar. Okay, so this is just a quick primer on interpreting the phylogeny. Okay, so in the next slide you will see different plant families that we've highlighted, and what we are looking for are patterns of cultural convergence. So, which we defined as different cultures converging or using related plants, evolutionary related plants for the same medicinal applications. Okay? And the reason why this is interesting is because we think there must be something in the phytochemistry and pharmacology of these plants for the different cultures to actually use them. So these different cultures independently discovered the plant's common uses, that family's uses. So this is the phylogeny that we generated in our paper. So this was published early last year in the Journal of Herbal Medicine. So this is in collaboration with Camila Javier, which I forgot to mention. She's pictured right there. Not this guy, this one. I don't know that guy. So this was published last year, and this is the phylogeny. So I apologize if some of the lines are a bit faint, but you could, I'll be happy to show you the paper later on. It did not come out nicely on the slide, but oh yes, and even the different plant families. So I actually also shaded the different plant families which we found patterns of cultural convergence. So again, we're looking for different cultures using related plants for the same medicinal application. So we found 10 plant families that showed that pattern that we're looking for. Lauraceae, Zingiberaceae, Apiaceae. So Lauraceae is that family that I presented a while ago. The bay leaf family, Zingiberaceae, the ginger family, Apiaceae, carrot, fennel family, celery. So these 3 families are being used for gastrointestinal problems. Okay, so you can see right here. And also for other things, but notably for gastrointestinal problems. And in the later slides, I will explain why this is. And then Lamiaceae, which is the mint family, we found 3 out of the 5 cultures using it for gastrointestinal problems as well as respiratory problems. Rubiaceae, the coffee family, and Meliaceae, which is the neem family, we find different cultures using them for antibiotic uses. Okay. And another family that's interesting is Burseraceae, which is the myrrh or frankincense family. We find that 4 out of the 5 cultures we surveyed use it for musculoskeletal problems. Okay? So I won't be able to talk about each of these plant families in detail. I'll present a couple of examples that will be Lamiaceae, the mint family, and Burseraceae, the myrrh family, and I will also try to explain what could be the phytochemistry and the pharmacology behind this pattern that we're seeing. So in the mint family, we found 3 out of 5 cultures using it for gastrointestinal problems. So this dark blue, and for respiratory problems. And so what we did, so we were fascinated by this pattern. So what we did was go into PubMed, look at the literature, try to find what main phytochemicals in the mint family that may be important for these medicinal applications. And we found that monoterpenes, terpenoids, which are responsible for the aromatic volatile oils, the essential oils in in members of this plant family, we found that they have many physiological functions. Carvacrol, for instance, which is a monoterpene found in many members of the family, strongly anti-inflammatory. So it makes sense that people, these different cultures would use it for alleviating gastrointestinal problems. So it has a carminative application, so removing flatulence, alleviating flatulence. It is spasmolytic as well. Okay? Another monoterpene that we found, menthol. Okay, so this is also used for alleviating decongestion, and that could also explain why it is being used for respiratory problems. So in general, for this plant family, the presence of monoterpenes and terpenoids are important for these therapeutic areas for gastrointestinal and respiratory problems. Another plant family that shows a pattern of cultural convergence. So we have here 4 out of the 5 different cultures we surveyed. So you see the 4 different colors here. So these are the different species within the Myrrh family, Burseraceae. And these different cultures consistently use species of this family for alleviating musculoskeletal problems. So we found that to be super interesting. And so we went into the literature, tried to find what's causing this pattern. And what we found was that this family is rich in triterpene acids called boswellic acids, and they have anti-inflammatory and hyperanalgesic properties. So it's strongly analgesic. And in fact, there was this American company based in Indiana that already prepared an extract from the resin. So the resin from one species, from Boswellia serrata, and they are marketing it for osteoarthritis as well as for improving joint pain. And it has undergone clinical as well as pharmacokinetic studies and has been proven to be very effective. So this is again one of those examples where you derive a medicine from traditional uses. Another thing that was very interesting in our study, so one plant family that was highlighted to show pattern of cultural convergence is Apiaceae. So this is the carrot, dill, fennel, parsley family. These are the different species within that family, and you can see again different cultures using the different species within that plant family. And you can see that majority of the species again have gastrointestinal uses. Okay? However, there were 2 species, Angelica sinensis or Dong Quai, which is used in Chinese medicine, and Ami bisnaga, also known as bisnaga or Kella in Middle Eastern medicine. These 2 species we did not find any reports of traditional uses for gastrointestinal problems. But because we know that other species within that plant family are used for that, we looked into the literature and tried to see if we can find experimental evidence for these 2 species, for Dong Quai and Bisnaga. If we can find experimental evidence that they have gastroprotective phytochemicals. And lo and behold, we did find a couple of studies that tested the extracts of these plants, and they found that they have gastroprotective properties. So they have phytochemicals that have gastroprotective properties. So what we're trying to show here is that the phylogeny was a very important predictive tool in allowing us to discover new medicinal applications for these two species. Okay? Because we know that related species also have those uses. And if you look at other medicinal applications as well, there are also other interesting phytochemistry and pharmacology that can be explored for these other uses, but it was rather difficult to find information in PubMed, so probably no one has really worked on it. There was a review paper that came out in 2011 on the phytochemistry of this plant family, and the authors are saying that aliphatic polyacetylene, so long-chain hydrocarbons, within this plant family, so it's rife within this plant family, and they have many physiological functions: anti-inflammatory, antibiotic, anti-parasitic, antifungal, even for appetite suppressant properties, things like that. So it has very interesting pharmacology and phytochemistry that we should go explore. And before I move to the next slide, I forgot about this one. There was also, so we found 3 different species also being used for female reproductive concerns, so gynecological problems, so this pink boxes right here. And so we tried to also find information in PubMed if other species within Apiaceae also have phytochemicals that may be important for this. And again, we found evidence for Centella asiatica, for Gotocola in Indian medicine, and for the Chinese herbal medicine or species Bupleurum chinense. We did find that they contain phytosterols, progesterone and estrogen mimics that we know can affect female reproductive function. So again, these 2 examples indicate that the phylogeny is a very important predictive tool that would allow us to discover new medicinal applications. And so in conclusion for this first study, we did find patterns of cultural convergence. We found 10 plant families where you have different cultural groups using related species within that plant family for the same medicinal application. Of course, we weren't able to survey all of the herbal species that are found in New York City, but I think the nice thing about phylogenetic analysis here is that you can keep on adding new medicinal species, and that will only corroborate the patterns of cultural convergence. So it will add more species to a particular plant family and even highlight more families that are potentially therapeutic as well, say for male reproductive concerns or for endocrine problems, diabetes, cancer, things like that. So you can just keep on adding on the phylogeny to find more patterns of cultural convergence. And lastly, we saw that the phylogeny is a very important predictive tool in allowing us to uncover new medicinal applications as well as new drug sources. So in the next study that I will talk about, and I think the primary reason why I am here, is our study. It's a very similar study to the first one, but this time we just focused on psychoactive plants. And we also, this is also at the global scale, so not just restricted to New York City. So you can think of it as sort of like a review. And this was in collaboration with this my former grad student Nashmaya, who was very interested in psychoactive plants. So our questions for this paper, so this was published last year, and so we asked if there's a similar pattern of cultural convergence in psychoactive plants. Are there certain plant families that are predominantly used as hallucinogens, as sedatives, as analgesics, and so forth? And then in addition to that, we also ask if there is a pattern of mechanistic convergence, wherein unrelated species that have the same psychoactive effect, so let's say as hallucinogens, do they affect the same neurotransmitter system? Okay, so these were our 2 questions. And like the first study, we tackled these questions by reconstructing a molecular phylogeny of different species. So we compiled a list of culturally important psychoactive plants. We had 120 plus, at least 120 genera, psychoactive genera. So our main reference for this was Christian Rach Encyclopedia of Psychoactive Plants, but we definitely also looked at the Schultes and Hoffman book, as well as other peer-reviewed papers. So similarly, we obtained the RBCL gene for these different species to reconstruct this phylogeny. And I know it looks very big and convoluted, but I promise that I will break it down in the following slides. But for some general patterns, okay, so like the first study, we also indicated on the branches the different cultures that used them. So for this study, we identified Native American psychoactive plants. So that's indicated with red branches. Okay. When we say Native American here, we mean psychoactive plants from North, Central, and South America. And 49 out of the 120 plants that we surveyed actually came from that region. We also have European culture represented here, which we indicated with blue branches. So those plants that have blue branches next to them, that means that it is used in European culture. We have Middle Eastern. Those would be represented by the orange dashes. And I am sorry if can you see that it is orange? I'm not making it up, it is orange. Middle Eastern and African culture, we merged them together because of their geographic proximity. So that's indicated by the orange dashes. We also have temperate Asian, which is the pink lines right here. Temperate Asia would include Russia and countries in East Asia, Japan, China, Korea. We also have Indo-Malayan psychoactive plants, and we define Indo-Malaya as India plus Southeast Asia. And then finally, we also have a few plants from Australasia. So we define this as Australia and New Guinea, represented by yellow branches. And we really just had a few, I think 4 out of the 120 genera. So this is a genus level phylogeny, not species level. And the reason is there are many species, so there are certain genera that have many species. Okay, so that we don't bias the phylogeny, visually bias the phylogeny on these genera that have many species. We decided to make a genus level phylogeny. And on the right of the plants, we have the different psychoactive effects. So the first column here, we have plants that are being used as hallucinogens in their respective culture. Okay, so the red boxes are those that are used as hallucinogens. The green boxes, sometimes it's not working. Okay, the green boxes represent plants that are used as stimulants. The pink boxes represent plants that are used as anxiolytics. So we also define anything that's used for relaxation, as calming agents, as anxiolytics. So they fall under this category. So they are indicated with pink boxes. And then the blue boxes, those that are used as antidepressants. So we find that phrase, that term is not often used, antidepressant. So any description, any traditional description that we see that says uplifts the mood or spirit, we define that as antidepressant, and so we indicated that with blue boxes. Then we have sedatives. Those that are sedatives are in gray boxes. And then additionally, we found that many psychoactive plants have analgesic and aphrodisiac effects. And so we also indicated that on the phylogeny. So analgesic effects are represented by the orange boxes, and then aphrodisiac effects are represented by the yellow boxes. And then to answer our second question about mechanistic convergence, so if unrelated plants that work similarly have the same psychoactive effect, do they affect the same neurotransmitter system? So what we did to answer this question was again look at the literature. We first determined the main phytochemistry in that plant family, and then we tried to infer the neurotransmitters that are being affected, also based on literature. And after listening to 2 days of talks, I understand that what I have presented here, what we have presented here on the paper, like in terms of neurotransmitters, it's overly simplistic, and I would like to apologize for that. But we are seeing patterns here. So I'm hoping that you could also see the general patterns. So I will discuss that later on. You don't see any red thing, right? Okay. Yes, it's not working. So I will discuss that later on. So just as a general overview of this big phylogeny, what we found that, don't we have any other pointer? Because I don't, oh yes, here, okay, so it's working. So here, Myristicaceae, the nutmeg family, Fabaceae, bean family, Cactaceae, cactus family, Convolvulaceae, the morning glory family, and Solanaceae, the belladonna family. So they are predominantly used as hallucinogens by various cultures. Okay, so those show patterns of cultural convergence. And then we have here Papaveraceae, the opium poppy family, and Lamiaceae, the mint family, being used as sedatives and analgesics. Okay, and then Solanaceae here, the belladonna family, used as a sedative, not necessarily as an analgesic. The mint family, again, Lamiaceae, have this additional use as an anxiolytic. And then we also found that the unrelated families, Malvaceae, which is your cacao family, and cola nut family, and Rubiaceae, the coffee family, and kratom family. Those are stimulants. And then we found that Apocynaceae, many cultures use it for uplifting the spirit. So we indicated that to be antidepressant. And then finally, 3 plant families, the Asteraceae, sunflower family, Solanaceae again, and Apocynaceae again, the milkweed family, where ibuga belongs. Those were also indicated to be aphrodisiacs. So in the following slides, I will go through each of these psychoactive effects and these families and explain what could be the phytochemistry that is underlying these patterns. There's this other figure in our paper wherein we showed the cultural distribution of the various psychoactive applications. So going this way, row by row, you will see the distribution of the different psychoactive plants within each culture. So this is Australasia, China, Temperate Asia, Indo-Malaya, and so forth. So the different colors refer to the different cultures. So you would see that the red bars, those are for Native American culture. You can see that most of the plants, psychoactive plants they use are primarily hallucinogens. Okay, so it seems like hallucinogenic use is one of the preferred the main preferred psychoactive effect for many of these plants. And then another important psychoactive effect that is sought for is the sedating effect. So you see these peaks right here. So many psychoactive plants in different cultures are used as a sedative. So hallucinogenic and as sedatives. So we thought that's a very interesting pattern. So with regards to the plant families that are used as hallucinogens, so again we have 5 different plant families presented here. By the way, you would see next to the plant families there are pie charts or more of gray circles next to them. This just indicates the generic diversity of that plant family. So it is proportional to the number of genera within that plant family. And the reason why we did this to show that the patterns that we are seeing for many psychoactive families is not an artifact of the generic diversity of that plant family, except perhaps for Fabaceae, but for other plant families like Myristicaceae, only has 20 genera, but it's still highlighted as a psychoactive family. Okay? So you will see pie charts or circles again in the following slides. So the nutmeg family used by different cultures, as you can see here again, different colors right there, as hallucinogens. The bean family, so we've been talking about the bean family, at least the individual species or the individual genera or genus. You have, we've talked about mimosa, acacia, sophora. These are represented here, so they are primarily used as hallucinogens by different cultures, but primarily by Native Americans. Cactus family, only Native Americans seem to use this family. So you can see that it's all red right here. But they use different for the hallucinogenic effect. And then morning glory family, again, different cultures. And then Solanaceae, again, different cultures indicated by the different colors used as hallucinogens. And so what could be the phytochemistry and pharmacology explaining these patterns? So in Myristicaceae, nutmeg, we found myristicin, elemicin. These tend to be serotonin agonists. Okay, so they bind to serotonin receptors. So we think that this is responsible for that hallucinogenic effect. In the bean family, it was mentioned like dimethyltryptamine, DMT, and other tryptamines, bufotenin, mucuna itself right here produces serotonin. Again, serotonin agonists. The cactus family, mescaline is another serotonin agonist. There's also probably other phytochemicals that are unexplored in other species that also work the same way. For convolvulaceae, ergot alkaloids, as mentioned yesterday, Dr. Nichols, yes, Dr. Nichols, also serotonin agonist. However, for Solanaceae, it seems to exert its hallucinogenic effect in a different way. We find that the propane alkaloids are acetylcholine inhibitors. So when it's shaded red like this, that means it's an inhibitor. On my slide, these should have green background, so it means that their levels are increased in the presence of these phytochemicals, but the green background is not showing. But whenever you see the red background, that means it's an inhibitor. So the terpene alkaloids in Solanaceae in general inhibit acetylcholine. And these plant families here are unrelated. So they are found in different parts of the phylogeny. But you can see that they seem to work similarly. So they are used by different cultures as hallucinogens, and they all work as serotonin agonists. And again, this might be overly simplistic, but I think we're seeing some patterns. For Solanaceae, so the inhibition of acetylcholine can also produce a sedating effect, a relaxing effect. So we know that acetylcholine is responsible for muscle activity, so to inhibit that would also cause a relaxing effect. So it's more of a calming, I guess, hallucinogen. But I've never tried any of these, so I can't really tell you. And then another interesting thing, we found that among the hallucinogenic asterids, so when you look at the phylogeny of flowering plants, there are two main groups, the rosids and the asterids. So just to quickly just show you that, here you have rosids and asterids right here. So there seemed to be a bias on the use of asterids as psychoactives. We don't really know the explanation for that. But more than 50% of the hallucinogenic asterids, we found that they are also used as aphrodisiacs. We don't really know why. It was hard to find papers. But again, just looking at the neurotransmitters that are generally affected for Asteraceae, so there seemed to be direct correlation between the hallucinogenic use and the aphrodisiac use. We found information that Asteraceae is rich in sesquiterpene lactones, but we cannot pinpoint the pharmacological effects and the physiological effects, so that's why we have question mark right here. We don't really know if those sesquiterpene lactones are responsible for the aphrodisiac effect. For Solanaceae, again, acetylcholine inhibition. This might have something to do with the aphrodisiac effect. We were able to find reference that says inhibition of acetylcholine transmission can actually increase dopamine. And we know dopamine is a neurotransmitter involved in pleasurable sensations, sexual arousal, things like that. So maybe, but I don't know. So I can only speculate here. For Apocynaceae and Rubiaceae, so this is two closely related families within the order Jansenalis. So again, we find that many of the species are being used as aphrodisiac. Some of these are hallucinogens. We don't really know if there's a correlation. It might have something to do with the increase in the synaptic levels of these three neurotransmitters, but I don't really know. So this is something to explore.
I lost track, actually.
Okay. Yes, I'll be done in 10 minutes. Sorry.
Every word here.
Okay. So for those plant families that are being used as sedatives, we found three unrelated families. Again, the opium poppy family, Papaveraceae. We find not only papaver, papaver is the genus of opium poppy, but we find other species within that family. So Argemoni, Estrolcia being used in by Native Americans as sedatives, and then Meconopsis used by temperate Asians as sedatives. So again, this beautiful pattern of cultural convergence. Of course, we know that these different cultures did not exchange information, right? I mean, they independently discovered that that members of Papaveraceae can be used as sedatives. So at that time, they didn't have Facebook or Twitter to exchange this information. So I think that's a very interesting pattern. Solanaceae, as I said, not only used as hallucinogens, but also as sedatives. So many species can be used as sedatives. As you can see by these gray boxes, so there are other species that are not used as sedatives. Nicotiana is one because it is a stimulant, so it's not used as a sedative. And then for Lamiaceae, also we have different cultures using it as sedative. So what could be the phytochemistry and the pharmacology explaining this? So in the opium poppy family, we know that isoquinoline alkaloids like morphine, codeine, thebaine, those bind to opioid receptors and in doing so produce that sedating effect. We don't really know what's going on in other species, but it's possibly similar alkaloids. There are similar alkaloids that are possibly present as well. For Solanaceae, I've already mentioned that inhibition of acetylcholine can produce that sedating effect. And for Lamiaceae, it works on a different mechanism. So it works on the GABA neurotransmitter. So the phytochemicals in Lamiaceae increase GABA, and GABA is the main inhibitory neurotransmitter. So gamma-aminobutyric acid. So increasing that in the body would produce that sedative effect. And so we find here 3 different families having the same psychoactive effect with different mechanisms of action. In addition to that, we found that members of Papaver and Lamiaceae are also used as analgesic, so those orange boxes. So again, it may have something to do with the opioid receptors in Papaver, and in Lamiaceae also has something to do with the GABA receptor. So we were able to find information, papers on that GABA agonists can also have analgesic effects. And in addition to that, there's also an anxiolytic effect in members of Lamiaceae. And we were able to find information that GABA, again, also has anxiolytic properties. In fact, pharmaceutical anxiolytics like Klonopin, diazepam, they all increase GABA. Okay, so this family might be worthwhile for looking for further exploration of new drugs that have anxiolytic, analgesic, as well as sedative effects. And then for plant families that are used as stimulants, so we found these unrelated families, Malvaceae, the cacao family, the cola nut family, and Rubiaceae, the coffee family, and Mitragina or Kratom in this family. We found that they work on similar mechanisms, even if they are totally separated in the phylogeny. They are not at all closely related. So alkaloids within these families work as adenosine inhibitors. So adenosine is naturally sedating in the body. So the alkaloids present in these families prevent binding of adenosine, and that can produce the stimulating effect. And another mechanism by which these alkaloids work is increasing noradrenaline, so which we know is involved in the fight and flight response. Okay. And again, another interesting pattern that we found for those within the order Gentianalis, so Rubiaceae and Apocynaceae are closely related families within this order. We find that in addition to those that are being used as stimulants, many of them have traditional uses as antidepressants, uplifts the mood or spirit. Okay? We try to find information on what is causing this pattern. It might have something to do with the indole alkaloids, increasing synaptic levels of these neurotransmitters, serotonin, dopamine, noradrenaline. For some indole alkaloids present in these families, they also prevent reuptake of these neurotransmitters or decomposition of these neurotransmitters, which is the same mechanism of action that we find in pharmaceutical antidepressants. So pharmaceutical antidepressants prevent reuptake or decomposition of these neurotransmitters. However, one thing that's very interesting is that we know pharmaceutical antidepressants like Zoloft or Paxil, one of its major side effects is sexual dysfunction. But then when we look at these plant-based antidepressants, they have been reported to be used as aphrodisiacs. So what is driving that pattern? I don't know. I honestly don't know. I'm just thinking it might have something to do with dopamine. So this tells us that this order right here might be a good candidate for looking for antidepressants that don't have sexual side effects. And so, in conclusion, we did find, again, patterns of cultural convergence in psychoactive plants. There are certain plant families that are used as hallucinogens, and we saw that like the nutmeg family, morning glory family, cactus family, and so forth, they mostly work as serotonin agonists. Papaveracea, opium poppy family, mint family, used for sedation and analgesia, but they work on different mechanisms. Aposinaceae has antidepressant effects as well as aphrodisiac effects. And so we can explore these plant families as novel therapeutics for neurological disorders such as insomnia, depression, anxiety, etc., which reinforces the potential of plant psychoactives as springboards for psychotherapeutic drug discovery, as quoted from Dennis himself. Thank you so much for your attention.